Thank you, everyone. Our session has been graced by some great and eminent speakers. So, I would like to request our Honorable VC Ma'am and Dr. Navid to welcome our first guest, Dr. Navajit Chakrabarti, with the auspicious Tulsi saplings. I would again request VC Ma'am and Dr. Pooja to welcome our next guest, Dr. Manish Narang, with the Tulsi sapling. I would now request Dr. Navid and Dr. Pooja to welcome our VC Ma'am for gracing our occasion with the Tulsi sapling. Thank you, everyone. Now, ICEIL is a conference which aims at bringing together leading entrepreneurs, founders, serial entrepreneurs, academicians, researchers, professional innovators, management professionals in senior leadership, policy makers, and whatnot. They all are here to exchange and share information with all of us and to provide their research results, best practices, trends, innovations, concerns, challenges, opportunities in all aspects of entrepreneurship, innovation, and leadership. So for this purpose, I would now request Dr. Tanveer Naved to provide a welcome address to our audience. Dr. Naved.
Very good morning to one and all. Honorable Vice Chancellor, Madam, the esteemed speaker of the day, and my colleague Dr. Pooja and Dr. Puniti. It gives me immense pleasure to welcome you all to this workshop on computational drug discovery, bench to market. This workshop is being part of a mega event, the 5th edition of International Conference on Entrepreneurship, Innovation and Leadership, which will commence from till the 16th of this month. I firstly like to welcome our Honorable Vice Chancellor, Madam Dr. Balwinder Shukla, Madam, to grace this occasion. And the whole concept of this international conference is the brainchild of Madam. We organized the first ICL 5 years ago, and ever since, the stature of the conference is getting bigger and better every year, thanks to the wonderful leadership of Madam. I also extend a very warm welcome to our esteemed speaker, Dr. Navajit Chakrabarti, the head of Bio-IT Radcliffe Genetics. Welcome, sir. And also Dr. Manish Narang, Director Medical Affairs about Vascular. Welcome, sir. Dr. Avinash Mishra, the CEO of Grovdia Technology. Another speaker will be joining very shortly. I also welcome my colleague Dr. V. Pooja, the officiating head of AIP, and also Dr. Puniti Mathur, the deputy head of AIP, And I extend a very warm welcome to all the participants, especially outside from MIT. Madam, I am very pleased to inform that not only from MIT, but we got the overwhelming response from the outside MIT also. And we have the participation from all the reputed institutions of NCR, which include Jamia Hamdard, Delhi Pharmaceutical Research University, Sharda University, Galgotias University, Arvind Lawland, Noida Institute of Technology, RKGIT, ITS College of Pharmacy, Metro College of Pharmacy. So these are the number of institutions participating in this workshop. And I recall my graduation days some 2 decades ago, when all the disruptive technology were very lesser known. But in this modern era, I think bioinformatics, computational drug discovery has a very pivotal role to play, especially in the pharmaceutical and biotechnology sector. And I'm sure the scientific deliberation today will be very beneficial to all the students. With this, I welcome you once again. Thank you very much.
Thank you so much, sir, for your kind words. I would now like to request Dr. V. Pooja, who is the officiating director, Amity Institute of Biotechnology, to please grace us with few words. Ma'am.
Thank you. First of all, I would like to welcome all our esteemed guests and our Honorable Vice Chancellor on this dais. It is our Honorable Vice Chancellor's brainchild when she first, started International Conference on Entrepreneurship, Innovation and Leadership. Way back in, I think, 2018, this is the 5th International Conference, and as Dr. Naved said, year after year, it is only becoming stronger, and it has grown so much that now students and guests from all over the country and even from outside India are participating in this conference. So as a pre-conference, this computational drug discovery bench to market was envisaged, and I'm very thankful to Dr. Naved, Dr. Kamal Rawal and their team who has made an effort for this workshop. As you all know, bioinformatics is the backbone of drug discovery. The mankind could be saved only because of the interventions of bioinformatics, where repurposing of drugs was carried out. Today we hear so much about resistance and antimicrobial resistance and new pathogens which are every day arising. Every day you have a new mutation that is happening in these pathogens. So the drug discovery cannot be a 20-year process. That we have learned from COVID, that it cannot be a process which takes 20 years for a drug to come into the market. And that is the reason why we need bioinformatics and computational biology interventions. Because if you do not start finding out new drugs from the plethora of compounds that you have, with you, millions of compounds which are already present, which are already approved, or which are already approved from some other disease, if you do not repurpose them for the new diseases, then again billions of dollars and 20 years would be the time that would take for these drugs to come into the market, and that would play havoc to the mankind. Obviously, as I said, all pharmaceutical industry, biotech industry, all of them now have a very dedicated bioinformatics cell. And as somebody who has been a part of, placements and Industry Connect, I know what is the requirement of the students who are well-versed with computational biology, with new drug discovery, target discovery, AI, ML, big data. There is a huge, requirement in the market. Probably 15 years ago, there were not more than 5 or 6 companies which started working in this area. 7-8 years ago, probably pre-COVID, there were probably 50 startups in the country. Today, we have 6,500 startups in the country, because bioinformatics requires least input of resources. You require one computer and your brain, and you can start a company from a coffee shop. And you can get projects from around the globe. So that is the beauty of bioinformatics, and this workshop has come at a very, very apt time, because just now we got a message that internship for 2025 students, 2025 pass-out students who have computational biology understanding, AI ML understanding, and the internship salary is 1,50,000. It is a global company. So if you start learning these techniques, if you become skilled and competent in these techniques, you will work for global multinationals. So please do not take this lightly. We had 5-7 students coming into bioinformatics course. Today we have 70 to 100 students coming into the bioinformatics course. So that is how bioinformatics is growing, and we are one of the very few universities which give undergrad degree in bioinformatics. Take your subject seriously, and this is a very important conference. So all the best to all of you who have registered. Congratulations that you have registered, and congratulations that you would be learning something which would help you for your lifetime. So all the best, and once again, thank you so much.
Thank you, ma'am, and a big congratulations on such a good news. We all are here and have gathered for um, this workshop, and we have with us Honorable VC Madam, who is an institution in herself. Ma'am is a PhD from Queen's University, UK. She has over 3 decades of rich scholarly experience in industry as well as academia. Dr. Shukla is a professor of entrepreneurship, leader, and IT, and the vice chancellor of Amity University, Uttar Pradesh. She has been one of the key team members, spearheading and instrumental in the exponential growth of Amity Group engaged in higher education. Ma'am, who has been a guiding light and a source of continuous inspiration, is an excellent administrator who leads by example. May I now request Professor Dr. Balwinder Shukla, who is the conference chair and the VC Amity University, to please grace us with few words.
A very distinguished guest, Dr. Navji Chakravarti, Head of BioIT Redcliffe Genetics, Dr. Avinash Mishra, CEO Grow Idea Technology Private Limited, who will be joining us shortly, and Dr. Manish Narang, Director, Medical Officer about Vascular, and who is present here, and also Dr. Sudarshan Pandya, Principal Scientist, Schrodinger, Bangalore, who is present online, and my colleagues on the dais, Professor Naved, our Deputy Dean, Health and Life Sciences, Dr. Pooja, Head of Amity Institute of Biotechnology, and Dr. Puniti, Deputy Head of Amity Institute of Biotechnology, my colleagues in the audience, but most important, future leaders in healthcare, biotechnology sector. First of all, I would like to extend very warm and hearty welcome to all the distinguished speakers, participants to this workshop, which is a pre-conference workshop. The idea of starting the conference on entrepreneurship, innovation, and leadership was, you could see the very clear writing on the wall: the future belongs to innovators, future belongs to entrepreneurs, and the future belongs to leaders. And how can we bring these 3 aspects together under one umbrella? That's how this conference was conceived. The technology is playing the most important part, and we have seen there used to be the industrial revolution. After one industrial revolution, you have the century or more to have the next industrial revolution. Then you have the couple of decades, the third industrial revolution, and then the couple of decades, the 4. Now, today nobody talks about the decades for the next industrial revolution to arrive. And it's only the technological developments. And I remember in 2018 when we said very proudly the education 4.0 based on the technologies of the industry 4.0. And in no time, the next set of the disruptive technologies came and we started calling it the industry 5.0. And almost 6 months back, our Honorable Prime Minister was in one of his address was saying the industry 6.0. And now when we are having the G20 and where the technologies of the sixth generation, are there and they're talking that how the future disruptive technologies are going to play a very important role. We have seen with the help of the technologies, and we reached to the moon through the Chandrayaan-3, a great success. The world has praised the leadership of India, the technological developments by the India, and we were not able to sync even with the success of Chandrayaan-3, and we saw the Aditya L1 also launched. And it's just about, not even the 2 weeks, and we are now hearing about the Gaganyaan also. So when we talk about the technology, it's not only the IT technology. It is the material sciences. You will look at the hundreds of technologies come together. Materials to devices to the communication to all those things to ultimately make a mission for the success. And when we are looking at the future of the nation, one side, the technologies are important, but the areas where we look at agriculture, healthcare, the environment, the space, all those things, the defense. But I think the health comes at the number one. कहते हैं कि health है तो wealth है. So how you really take care of the health. And as Dr. Pooja was saying, that you will not wait for 20 years the new drug to come. When the COVID came, without knocking any doors, it arrived at our doorsteps. We had to close our companies, we had to close the academic institution, travel, whatnot, overnight. And then how to be connected with each other, how we progress our businesses, how we progress the education, how we progress, take care of our customers, how we maintain the supply chain, all again the technology played a very important role. That we are connected with each other, whether it was the Zoom or WebEx or MS Teams or Google Talk or whatever, and all of you have experienced that how your classes were conducted. And that was just very small example, but it connected the whole world, and we did not feel that pinch of not traveling. At least we were connected. The entertainment industry flourished like anything. When in shortest possible time, the vaccine was brought to the market. And the lakhs and lakhs of lives were saved. Imagine if these vaccines did not come, what would have happened to the world today? And we all realize the importance of taking care of health and working together. And that's why today we see in the healthcare one side how the technologies can help, uh, in the healthcare, not only the drug discovery, but also the personalized medicine, precision medicine, so many other areas. Another thing which played, realize that collaboration is again the need of the hour. You cannot work in isolation. You need to collaborate with the, between the academia and industry, between industry, and within the country and outside the countries. So you people are really, really fortunate, all of you who are sitting here, that the future belongs to all of you. And anywhere and everywhere, the world is looking at how this youth can play important role in innovation, solving the societal problems or solving the industry problems. So the various verticals, when we discuss the healthcare becomes one of the very important verticals. Agriculture becomes another very important. Environment, sanitation becomes, the water becomes very important vertical apart from the defense and the space when you look at. And recently MIT when hosted the Science 20 vertical of G20, and we have seen the success of G20, which has been only hosted by India this weekend, and how the world leaders have praised India's, the power, India's capacity and capability, where we have not only showed our culture, but we also saw the technological advancements of this country. So I'm sure this workshop, when we conceived with the idea, because conference will be having the 58 parallel sessions. So all of you are invited to attend any of the sessions as per your choice, because all the sessions are available on the website of the ICEIL 2023. And but organizing this workshop, pre-conference workshop, was because we consider the healthcare as extremely important. And the student must understand what are the developments are there, and especially when we talk about the use of the technology and information technology. So bioinformatics, healthcare, that becomes very important. So I'm sure all of you are going to listen very carefully to all the distinguished speakers who are present in person or online, because their words, their experience is invaluable, and you must take advantage of that. And today the kind of this learning is where it's not only the classroom learning which makes a person skilled, but it's attending the various kind of workshops, seminar, conferences, internships, apprenticeship. They also help in a big way to develop the knowledge and the skills and competencies to be ready for the profession, whichever the profession you choose. At MIT, we conduct the aspiration survey, as all of you know that who are with MIT. And then working through those aspirations, whether you're going for the corporate career, or starting your own venture, or going for higher education, or preparing for the competitive examination, how important is to understand the need of each career aspiration, how you prepare. But this kind of workshop prepare you for all kind of the career aspirations. So I would like to, again, on behalf of Honorable Founder President, Honorable Chancellor, all my deans, and on my personal behalf, extend a very warm welcome and thanks to our distinguished speaker for sparing your time and being here. As I know morning time is very, very important time, but being from away from your office and being on campus show that how keen you are to share your knowledge and help the youth, the future leaders, to develop themselves. And I'm extremely thankful to all of you, and I would like to congratulate Dr. Nave, Dr. Kamal, and the entire team of AIB for working on bringing such excellent speakers. And I'm sure it will be not only benefit for the student, but it's going to be beneficial for the institution to further discuss, collaborate for the various collaboration to work together in the area of research and innovation. Amity has been filed more than 2,000 patents and more than 30 technologies have been transferred to the industry, and we are looking further to work in the translational research and how we can work in partnership with the industry. So, and that definitely my colleagues will be exploring with you. And let's work together and solve the important problems of the society, where the industry can really serve the society in a big way in partnership with academic institutions. So thank you so very much, and I wish all the great learning to all the students. Wish you all the best.
Thank you so much, ma'am. We are now graced with our third speaker, Dr. Avinash Mishra, who is here. I would like to request our VC ma'am and Dr. Daved and Puniti ma'am to kindly give Tulsi sapling to him and welcome him. Thank you, ma'am. Thank you, everyone. This pre-conference workshop has been envisaged because we all understand how computational drug discovery is an effective strategy for accelerating and economizing drug discovery and development process. To talk more upon this and to tell you more about the workshop, may I please call upon stage Dr. Dr. Puniti Mathur, who is the Deputy Officiating Director, Center for Computational Biology and Bioinformatics, Amity University. Ma'am, please, could you just let us know more about this?
Thank you very much, Dr. Rennie. A warm welcome to all of you, all the participants. And at the outset, I would congratulate Dr. Nawed, Dr. Kamal, Dr. Abhishek for having this panel in front of us. And we have a very, very experienced panel today, students. And I would like to thank each one of you, all the panel members, to have taken up the time to come here. This computational drug discovery is very close to our hearts, especially at Bioinformatics at Amity Institute of Biotechnology. We have a number of faculty members, more than around 18 faculty members at biotechnology who are working in the area of computational drug discovery. So our students of bioinformatics get a lot of courses on this particular topic, but not only biotechnology and bioinformatics, we have students in pharmacy, students in forensic sciences, faculty members in these institutes who are also involved in doing computational drug discovery. So, students, this platform of a pre-conference workshop gives you an introduction to what is computational drug discovery. And I would like to apprise each of our panel members as well as our participants from outside Amity that we work extensively on Schrodinger. Other than the other software that are available, Doc and all, Autodoc and all that we all are aware of, we also train our students on Schrodinger. So our master's students, our bachelor students who are doing internships with us work on this software extensively so that they understand how to do it. So this conference is open to all, and the idea is that you all get an introduction to what computational drug discovery is, what small molecule drug discovery is, and how these platforms help you to accelerate the time period of drug discovery. So we have we'll have with us skilled personnel from Schrodinger who are going to take this session on Mastro, that's the interface of Schrodinger, and they will introduce to various different methods of computational drug discovery, right from docking to virtual screening to pharmacophore design and dynamics. So what you're going to get is just an overview of the whole thing. The idea is that you get an insight into what this is, and slowly and steadily you work on it. Whatever interests you, you are always welcome to come to us and work further on computational drug discovery. So without taking too much time, because I feel we are already running behind schedule, so I would request the session to go on, but welcome to all the participants, and I'm sure that you're going to make a big use of this platform that has been made available to you. Thank you very much.
Thank you so much, ma'am. So we now begin our session, and for that we have Dr. Navajit Chakrabarti. He has an exhaustive experience of more than 15 years of experience in bioinformatics, leading the bioinformatics team at Redcliffe Genetics and taking care of the bioinformatic operations and deliverables. Sir is an esteemed alumni of Regional Center of Biotechnology. He deals in bioinformatic analysis, next generation sequence data processing, RNA data analysis, hybrid assemblies, and other high-end technologies. He's highly skilled in data analysis of the sequenced data from first, second, and third year generations. Instrumental in handling multiple research projects and various collaborations with scientists across the country and globally. Sir, I would like to invite you for initiating this session.
Hello.
Good morning, all of you. So, I am here to give a talk on pharmacogenomics. So we are very much well aware about the pharmacogenomics and the application of genomics in multi-era generation. So as we know, pharmacogenomics is a field of studying that combine pharmacology and the genomics to understand how the genetic variation can influence a person's response to drugs. So genomics plays a very crucial role in drug discovery and development by providing valuable insights in the genetic basis of disease, how an individual can respond to a drug. So we all know about the human genome and the starting of the sequencing. So sequencing have a very pivotal role, played a very pivotal role in the sequence the human genome. And now we can say human genome act a pivotal role along the genomic develop a new area of research. So the unmatched and the scalability and the speed of the sequencer have given a dynamic era where we can sequence anything if we as respect to a disease or drug discovery. So the role of pharmacogenomics in drug discovery. So we can have the multi-era genome era where we can study the genomics, transcriptomics, metabolomics, proteomics to understand a disease, to understand how an individual responds to a drug. So we can there are different type of pharmacogenomics, like we can have a structured pharmacogenomics and we can have the functional pharmacogenomics. So first of all, we want to understand a gene which is having the role. Second, we can understand the variation in respect to SNPs, indels, CNVs, and we understand the linkage analysis like the phenotype data which is having the linkage with the mutation of an individual. We also study the gene expression, like how the expression pattern or expression profiling of an individual plays an important role in the drug discovery. So this is a NGS in how different applications of NGS are related. So we can, if we wanted to study the genome, we can go with that whole genome sequencing. If we wanted to study only the exome part of a individual, we can go for a exome sequencing. In the same time, we wanted to know about RNA, we can go with the RNA sequencing. We wanted to study the SNPs, indels, we will go for a reference-based whole genome sequencing to study the mutation in the individual. And so these are the different applications of NGS which will be having a role in the biological research. So whole genome sequencing, so what we wanted to study with using whole genome sequencing is, so first of all we wanted to study the genetic profiling. So genetic profiling, how a individual is, the architecture of a genome is, like what is that variation as compared to the reference. So how many SNPs are there, how many indels are there, whether the SNPs are in the known location or it's a mutation which is not known to anybody earlier. So what is the copy number variation? So all this can be studied using the genetic profiling in using whole genome sequencing. So we can also, using whole genome sequencing, we can understand a gene. So a variant, if we are identify a variant, whether the variant lies in the gene or it lies in the upstream of the gene or downstream of a gene, can be studied and can be useful in the understanding the genetic variation associated with the drug metabolism. So there are lots of pharmacogenomic panels are available, which is rightly can be taken and can be sequenced for any individual. So these are the key aspects of NGS which can be useful in the pharmacogenomics. So we can also discover the novel variants. So any variant which is not reported anywhere in the public domain can also be reported. So that will be again an application which can be used. So then we can also having the genetic profile of an individual using all the variants which is reported, linked with any phenotypic data which is available. So clinical implementations are also there. So once a genome is sequenced or an exome is sequenced, we can have the genetic diversity of a individual. In the same time, if we have the knowledge of the phenotype data, that how the patient looks like, what are the different problems he have. So we can link those markers with the phenotype data to have an understanding of the clinical side of the genome. We can also study the population studies. So when there is a group of individual having a control and a group of an individual having the disease, we can compare them based on the genetic makeup of the individual. So then the role of RNA sequencing. So we were very much known about RNA sequencing, where we wanted to sequence the transcriptome entirely of an individual. So what transcriptome will give us? The transcriptome will give us the gene expression profiling. So how the genes are expressed in an individual. So there are two groups. One is the control group, one is the treatment group. So we wanted to know how the treatment group behave to a drug. So what are the genes which get expressed, upregulated or downregulated. Then identification of alternate splicing. So we are very much aware of the alternate splicing, which will give the different isoforms and the variation having the variant functions. So these are the applications which can be studied using transcriptome sequencing. And then identification of the novel transcript, definitely. So the novel transcript which is not been known earlier and give rise due to the alternate splicing in the genome. So the biomarker discovery. So again, the comparing the different transcriptome of an individual, we can understand what are the biomarkers which is linked to any disease as compared to control and the treatment. Then drug induced. So once the drug is given to any individual, what is the transcriptome profile of the individual? So that can also be studied using sequencing. So these then pharmacogenomic drug development. So what we can do is here, once the entire transcriptome is studied and we understood the genetic variation in the genome, we understood the RNA profiling of an individual. So these we understood different expression level of an individual as compared to a control, how it is behaving in when the drug is induced to the individual. So these are different keys scope which can be used for the study of the pharmacogenomics. Yeah, so now I am jumping to the bioinformatics portion, that how the data can be used, analyzed and interpreted. Okay, so we are very much aware that we talked about the bioinformatics pipelines. So pipelines development is very like sophisticated nowadays, because earlier the pipelines were like just it's an end-to-end work. Like you got the data, you got analyzed for the first step, second step, third step, and then you are getting the result out of it. So now the pipelines are very much we can say now we are working on a pipeline management system. So there are two very much popular pipeline management system are there. One is Nextflow. I think you might have aware of this, and second is SnakeMake. So these are completely pipeline management system where the pipelines can be integrated, pushed, and you can have the reproducibility of the same pipeline wherever you are running it. So it will give you the confidence of analyzing the same result again and again. So there will be no changes in the pipeline, or whether you are running here or you are running in other servers. So let's start with the whole genome sequencing. So once the data is out, raw data from a sequencer. So we are very much aware of the Illumina sequencers. I think everybody is aware of Illumina sequencer, right? No? So yeah, so once the data is out from Illumina sequencers, so we have to analyze the data to understand the variants in the data. Okay. So first of all, what we'll do is we will take the data, we will do some pre-processing of the data. So Illumina adapters were there in the data. We have to pre-process this. We have to align the data with the reference genome. We'll call the SNPs and indels in the data. So these are a workflow that can be easily implemented to understand the SNPs and the indel in the data set. So once the variants are identified, we have to filter the variant, because there will be lots of false positive in the data. So we have to apply confidence filters to understand the to filter the data, and having the some confidence level to be incorporated to understand the SNPs on high confidence. So once the variants are called, we have to annotate them. Like we have different type of annotations are there: synonymous mutation, non-synonymous mutation, whether the mutation lies in a gene or it lies in an intergenic position. So different type of locations we can understand in the mutation where they are lying in the genome. Then we go about whole exome sequencing. So we know that 1 or 2% of the total genome is exome only. Okay, and so there are 85% of disease causing variants are located in a coding region. So it is very important that we can study the exomes. So for that there are different pipelines. One is Genome Analysis Toolkit and BARSeq. So these are very much popular pipelines which will be used to analyze the exome sequencing data. So this is the pipeline workflow where you have the raw data. So first of all, you will do the quality trimming, quality checking of the data, followed by alignment with the reference genome. Then the removal of duplicates. Then again doing haplotype calling to understand a variant in the data. So these are different workflows you can understand from where we can analyze the whole genome sequencing data to understand the SNPs, indels and the CNVs in the data and again going for an annotation to understand where those SNPs are lying in the genome, whether they are lying on the gene, intergenic or different aspects of the genome. So then we go for a genome annotation. So there are different databases in public domain which can be compared with the variant which is been captured or identified in the genome, and then we understood whether those variants are known variants or they are some new variant which is coming from the population. So these information will only be understood when we compare the variant with the public databases like DB SNP, 1000 Genome or ESPs in NCBI. So one more thing we can go with after we understood that there is a mutation which is novel or which is having known to any public domain, we can go always with the genome-wide assessment studies where the phenotype data is there and you wanted to link with the variant which has been identified in the genome. So the variation which will be linked with the phenotype can be called as interpreted as okay, this is the phenotype characteristic we have, and we understood that there is a SNP and with a very high confidence which is not reported anywhere maybe, and then it will linked. So there will be linkage between a SNP and the phenotype data. So this study is known as the genome-wide assessment study. Now we wanted to jump to RNA-seq. So first whole genome is known. So we understood that okay, there is a one genome where there is SNPs, there is a indels, there is CNVs which is linked to any disease or not. First, second, whether what is the annotation of the SNPs or indels which is been studied, whether the SNP is in the genomic region or it is the intergenic region or is within the exome. So those things can be studied using whole genome sequencing. Now we are jumping to the RNA-seq. RNA-seq where we will be studying the transcriptome of an individual. Transcriptome means the RNA. So first we study the DNA where we understood okay, SNPs, indels and CNVs, annotation, everything. Now we are jumping to the RNA-seq where how the transcriptome of an individual looks like. Transcriptome means the transcriptome profiling, how the genes are expressing in one individual when compared to the other. So one there is a healthy individual, one there is a diseased individual. How the healthy individual genes are profiling are there, and when the individual is prone to disease, what is the profiling of the individual having the disease. So those comparison will help us to identify the genes which is having expression as compared to the control. So we are taking control as a healthy. So these are the pipelines again. So where we will be taking the raw data, and then the raw data will be quality trimmed, and then we will proceeding with the adapter removal and the quality trimming of the data. So these are the generic pipelines which can be used to study the transcriptome of an individual. So once the transcript data is pre-processed, again it will be aligned to the reference genome. There will always be a reference genome which will be compared with the transcriptome profiling, and then the genes will be quantified and expression profiling will be recorded, and then we can have the differential gene expression to understand what is the TGs which is upregulated or downregulated based on the when compared to the control. So there are two concepts of technical replicate and biological replicate. So when the same samples is sequenced 2 times, we can say it's a technical replicate. Biological replicate means there are 2 individuals which is sequenced twice. So that is a biological replicate. This will enhance the confidence level of a DEGs which is been identified using RNA-seq experiment. Yeah, so there are different terminologies like there are different tools for expression estimation, differential gene expression. We can go with the different tools like coupling. Then we can go with the features count, Callisto, that can be used for studying the expression profiling of an individual. There are different R packages like ERJA, TSEQ2 that can be used for the differential gene expression analysis between a control and a treatment group. So these are very much generic plot that you can see how an expression pattern of an individual looks like. So the upper portion, red upper portion is the upregulation gene, and lower is the downregulated gene, and the black tells about the genes which don't have any significance. So definitely there will be 3 categories always. Either a gene is upregulated with significance, either a gene is downregulated with significance, or there will be gene which don't have any significance, or they are just having in the individual, but they don't have any role in the regulation of the transcriptome. So there are different, once the transcriptomes are analyzed and annotated, we can go with the Go enrichment, Go ontology, so that we understood what are the enriched plots, what are the enriched terms they are in the genes, which can be again been studied for the downstream, so that we can understand the functionality of the genes which is having the regulation in the individual. Yeah, then pathways. Pathways are very important. So we know what pathways are having regulation, which pathways are having important role in any disease or any disease or any individual which we are studying under. So we understood the okay, there is some pathway which is having role, and then we understood the okay, these are the genes which having regulation to this pathway. So those genes can be having a very important role in understanding the regulation pattern of the pathways, whether the pathway is triggered or no, based on the control and the treatment. So this is cake analysis. So you can see this green highlighted portion. Those are the genes which is high regulated, upregulated as compared to the treatment. So I am just giving you a pictorial representation where we can understand how a pathway, entire pathway, we can understand. Okay, these genes are having a upregulation as compared to the control patient, so that we can understand. Okay, after giving the drug, or if a patient is having some disease, how the regulation pattern of the genome looks like. Then we can study about a small RNA. So we know small RNA are the small sequences in length, only 50 base pair, and they bind to the 3' UTRs of the gene. So the targets will be the miRNA will always target the genome to have the role, cut the role, or to have the regulating the role of any gene in the vicinity of it. So there are different pipelines again. So this is a workflow which tells about how the small RNA is being studied. So you can again adding the data. You can do a data QC step, removing the adapter sequences, again mapping the data to the BLAST RFM databases, calling the novel variant, novel small RNA as well as known small RNA. Once the novels are called, you have to understand the structure, how the structure of the novel miRNA looks like. So typically, if the structure is there, you can tell that is novel miRNA. Then we can predict about the correlation of miRNA with the mRNA data. What is the targets which is having get targeted with the miRNA, that can also be studied. So these important. So those all the three applications what we have discussed having the important role in understanding the genome profiling, or you can say the architecture of the genome of any individual before we move with the drug delivery or the target gene on the target gene. Yeah. So the multi-omics era. So we can see now we have studied about in this session we studied about the genomics, we studied about the exomes, we studied about the whole genome, we studied about the transcriptome, small RNA. So these are the different applications. Along with that, we can also study about the bisulfite sequencing, where we will understand how the methylation sites are there in the vicinity of a gene that is having a role in the regulation of the individual. So these are different multi-omics application which will be very important and crucial in the study of an individual for the pharmacogenomics. That's it. Thank you. Thank you.
About this 125-year-old activity. Yeah, and we have multiple.
Thank you so much, Dr. Chakravarti. I would like to request Dr. Naved and Dr. Kamal Rawal to please felicitate Dr. Chakravarti for his immensely important session.
We have devices, we have pharmaceutical diagnostics.
So if there is any R&D section, I believe the kind of work that we are doing is really very meaningful. And it's in New Friends Colony.
Yeah,
But
that's
there are
the
different locations.
our India headquarters overall.
The video you
Bas. Thank you so much, sir.
Many.
So, as a researcher myself, I have always thought that research and entrepreneurship doesn't go hand in hand. I always thought that these are two different domains. But here with us, we have someone who has busted this myth and has created new paradigm shift in amalgamating research and entrepreneurship together. We have with us Dr. Avinash Mishra, who is the CEO of Grodia Technologies Private Limited. He has obtained his PhD in bioinformatics from IIT Delhi, from where he continued doing his postdoc. He has worked as an Indo-Australian Gold Fellowship, Australian Gold Fellow in Griffith University, Brisbane. Sir has been an active researcher, and he, with his company, is currently focusing on the computational biology domain with specialization in drug discovery. His main aim is to facilitate experimental biologists with computational techniques to improve the quality of research. May I please call upon stage Dr. Avinash Mishra, sir, to please enlighten our students with this session.
Okay. Thank you for this wonderful introduction. And honestly saying, which one is working? Both. Okay. So honestly saying, I was a bit confused that what should I talk in this program? Whether I should take all of you, because all of you are students, so a real work hands-on session where you can work little bit, but I believe that, and I was expecting that maybe all of you may have your laptop and we can work a little bit, but that's also okay. That made my job a bit easier. Yeah, so this company Grodia Technology, we have started in 2018, and I started from IIT Delhi, and now we moved to Gurgaon. So the whole objective of this company is how computational biology can be used for supporting the experimentalists, those who are working especially in drug design. Okay. So and by the way, if you have any question in between, just be casual and just ask question if you have. So what I decided now, just few minutes back, that maybe instead of going for a hands-on, maybe some other day I will take the hands-on when you guys are also ready. This time I'll just take you to some of the slides or some of the known information, but with a different perspective. I would try to put my perspective in that, and if you agree, great. If you don't, just raise your hand. So I think the best is that if I go through the presentation, and in between I will jump to some of the things that I just wanted to show you that what exactly we do in drug discovery or computational drug discovery, because we talked about this many times in many ways, but it is always important for students to understand that
what exactly we are doing, right? So it should not be a black box. Docking, molecular dynamic simulation, we all know about it, but now you also have to understand that the things are gradually shifting towards machine learning, right? So we all know that. And the biggest question is what we do in machine learning. Okay, I don't know as a student how much understanding you have, that what are the things that a machine learning program can do for drug discovery. And that was the idea, that if I can show you something which you can also do it, definitely it's a programming. So first of all, my suggestion to all of you is that learn some programming language. Okay. Now the question is which one. So I started with C++, but that was a bit old. Now we have like Python and R programming. Okay. So once again, and I'll take this conversation bit technical, and I hope that would be much beneficial for you. So between R and Python, I would suggest that go for Python, because Python has much more machine learning components. Okay, but if you guys are working in genomics area, or if you want to go in genomics, NGS kind of, then you should be comfortable with R. Okay, so only drawback with R programming is that the machine learning components are not so rich. Okay, but it has machine learning. The one which I wanted to share or show to all of you was on R programming. Okay, now when we talk about drug, we always have like some definition in our mind that what exactly the drug is. But if you go and find out that what is the conventional, and whenever the time is up, just let me know. So yeah, so drug is when defined in general, it is called as anything other than your food which is taken and making some changes in your body. Okay, anything. Okay, so that is like the general definition of a drug. So which is not a part of your diet, which is not a part of your food. Okay, but when we call drug, or in a research when we talk about a drug, then we actually talking about pharmaceutical drug. Okay, so these pharmaceutical drugs are actually the drug compounds that we use for treating, curing, preventing from the diseases. Okay, now if you go and find out that what are the sources of the drug, then the two major source, in fact the major source is nature, of course, but we cannot rely on nature for all the time, right? And it is also, of course, about the quantity of the production. So that's why we have to come up to the lab, and we have to produce it in the lab with a optimum quality that we want. Okay, so that's why lab and nature goes together. Now when we computational biologists talk about drugs, so what we refer, so we say that a compound which is in general called ligand, that binds to a biological target, which is most of the time protein, and either activate, which is called agonist, or inactivate the protein function, is called drug in general. Okay, it is I mean not correct to call it as a drug, but let's take the liberty and call it as a drug. But yes, so that is our perception of defining the drug molecule. Now on a structure perspective, if you see that what is a drug, so this key and lock and key model, I mean you all have might have seen it in your textbook, that this has been like from my time it was there, so that the lock is considered as a protein, and your key is considered as a ligand, right? Now and but there is a limitation in that. So what is the limitation? That this is not flexible, right? So this is a very rigid kind of model, this lock and key, and the protein and ligand interaction is not that rigid. Okay, but just to make it simple, we consider that lock and key goes like drug and protein. In terms of the drug target, that is protein, most of the times you can see that there are different classification of protein which is coming in most of the times as a drug target. Now, when we want to discover a drug, there are, I mean, you guys are going to have your that project or something similar to that. So maybe some of you decide that I will do something on drug design um, or maybe similar to this. So these are the 3 steps. So the first step is that you need to identify your disease area, okay, which one you want to target, right? So once you identify the disease, let's say I identify tuberculosis, and then the next is that what are the biological target that we need to use in this research pipeline, okay? So and there are certain criteria for defining a biological target. So like for infectious disease, what you want, and like I generally expect answers, so but I believe that here it is not going to come. So so yeah, so for infectious disease, when we are targeting some foreign particles, so we just want to kill them, right? But for some other disease, let's say genetic disease, we don't want to kill ourselves, right? So then the criteria of defining a target or selecting a target changes, okay? But somehow, okay, I'm not going in that deep, but yes, let's identify the target and then go to use your bioinformatics, cheminformatics, AtMed, machine learning, all those components, and try to design something which you have or somebody has not done it earlier. This is like the people used to do in drug discovery. It is a tedious process, lots of money, and that's why we don't have lots of drug discovery companies. Lots of money involved, time involved, expertise involved, okay? But slowly and gradually things are changing. Of course it is changing. Like this is my personal, based on my personal experience, I can say when I joined a BTech, there was a biotech boom, and I don't know where it is, but we heard about boom. But now I think that slowly and gradually it is going to come, and the market has been changed. And in fact, the Indian market also has been changed. Now you can find some bioinformatics company in NCR or in Bangalore or in Hyderabad, okay? My time it was not there. So it means that things are changing. It will take some time because it's a research component. So things will not happen in just one night, but of course things will definitely happen, and it is happening. So honestly, we also want to know that in drug discovery, when we say that it requires lots of money and it's very risky pathway, so whenever there is a risk, what we need to do is we need to analyze the whole process and try to mitigate the risk. This is what we do when I'm running my company, okay? So every time, for every process, there is a risk, and we need to identify that where are the factors that are causing a problem and how we can come up with a smart solution. And one of the solution is in silico drug design. And in silico drug design, we have structure based drug design and we have ligand based drug design, okay? So in ligand based drug design, we don't need the structure of a protein, okay? However, in target-based drug design, we need the structure of a protein. I think these things are pretty clear to all of you. So, but if you have any question, just go ahead and ask me that whatever your concerns are. This is like one of the picture of a target, how it looks like a protein. The helix, the strand, loops. Okay, so these are the different component of a protein target. And then the first thing is we need to identify that binding site. Like on a lock, we have a hole where we insert it. And this is like the whole of a protein where we want to insert it. But the thing is that there might be multiple sites on a protein which can accommodate your drug molecule. Okay, and so you need to understand that which site is actually the functional site. Okay, you may target the allosteric, but most of the times we go for the functional site. So that is very important to understand. So when you are identifying a cavity, just make sure that you are identifying the correct cavity. Definitely Schrodinger application is here, so you guys definitely can use it to do the virtual screening to identify the target of a binding site of a protein. Then you perform a molecular docking, so that gives you that, okay, this one is binding, this one is not binding. But believe me, the reality is that in computational program anything can bind. Okay, so don't trust blindly. Okay, so that is only my suggestion. So I'm not saying that the result is wrong, but you have to analyze the result before coming to the conclusion. Okay, so there are programs which can bind whatever you want wherever you want. Okay, so just like ChatGPT, you cannot trust 100%. Okay, so you have to read what is written by ChatGPT. So and then you calculate the binding energy. But once again, the binding energy is what is empirical scoring function. Okay, so because during my PhD I was designing these scoring functions. So I know that what are the limitation of a scoring function. So that might, you know, minus 11 kilocal per mole. It sounds fantastic, but yes, you have to investigate. You have to see that what kind of interaction it is making, how many hydrogen bonds you are seeing, how many stacking you are seeing. So based on that, you conclude. Now the things which I don't know you guys are doing it or not, molecular dynamics simulation. Desmond is a package in Schrodinger. We use Gromax. So once this molecular dynamics simulation has come, it has answered many questions like, can we trust on this protein and ligand binding? Is this interaction going to prevail? Or these kind of questions, okay? Because then the dynamicity introduced in drug design, okay? So that's like a brief of protein-based target designing, drug design. Now there is a ligand-based where okay, so I'll just come to this slide. So this ligand-based drug design is actually where the machine learning is directly going to impact, and it is being used in ligand-based drug design, because here there is something called QSAR, quantitative structure activity relationship. So there we use machine learning to establish a relation between a compound and its activity. Okay, so here it goes like, you first go and collect the data. What kind of data you want to collect? You want to collect experimental data. Okay, so the example is like if a class has 30 students. Okay, guys, whatever I'm saying, it's not written on the slide, so just listen. And if you remember this example anytime in machine learning, you will find it useful. So like suppose a class has 50 students like yours, and then there is a new guy or a girl coming and requesting to get admitted in the class. Okay, so generally what I mean, there should be some criteria of selecting that particular candidate for this particular class. Okay, it is not like ठीक है आ जाओ, right? और ऐसा नहीं होना चाहिए कि बस एक सीट खाली है अब आ जाओ, right? So there should be some criteria we need to follow, and how we come up with the criteria. Okay, just say once we go to the school for admission, so there is a test, right? So and that test is different for class 5th and class 10th, right? And somebody has designed this test, that okay, the candidate should know all these things, or a candidate, if a candidate will have these kind of attributes, then only he is perfect for this particular class. Okay, if no, then no, we cannot take it. Okay, so one way is based on my experience or teacher's experience, we make the assessment sheet. Okay, and then we assess the child. Other thing is, let's dig down the data. Let's go to the classroom and extract the attributes of all the existing students. Okay, and ask them few questions, and based on that you extract data. Okay, let me ask one question, that okay, how many times you go out for sports. Let's say this is the question. I'm collecting this data from 50 students and saying that okay, somebody is 2 hours, somebody 4 hours, and blah blah. Okay, similarly, how much time you are devoting to study. Somebody is saying 4 hours, because somebody is saying, but in general they all should be in some range, right? Otherwise somebody is exception, right? So then we collect all these features, and then we try to make a machine or just a prediction model, that if these features would be there in a new candidate, I will say yes. Okay, but one thing here, what you need is, you need also a negative data. You need a negative data. Okay, because there is one answer which is no. If you just collect the data from only positive data set, you won't be able to say no to anyone. Okay, so then what I will do is, I will go to some other class and collect the features. Okay, and suppose class 5 is my target, I go to class 8 and collect the features, and to all the members of class 5 I say 1, all the members of class 8 I say 0. Okay, and then train the model, classification model. Okay, and based on all the features, I will make a machine learning model, and once the new candidate would come, I would just ask those questions and just take their answers and feed it to the machine learning model, and then machine learning model will tell me that whether the guy should go to class 5 or not. Okay, the same thing we are doing in QSAR. We have bunch of known data set, and based on that we build a machine learning model, so that a new compound will come, I will able to classify whether it is a drug or a not drug. Okay, so that's a very I try to simplify it. That is we generally do, but this feature extraction is, and I would urge strongly to all the guys and girls that go and make yourself comfortable with machine learning. Okay, because maybe you are learning or reading stuff now, but maybe once you go to the market down the line, maybe 2 years, 3 years, I don't know. Okay, so by that time things will get changed. Okay, so I mean, ML is something which is going to capture most of the things in drug design or in computational biology. So if you guys are interested for computational biology, you should have a good understanding of ML. At the same time, a very good understanding of science. Okay, so because ML can give you sometimes rubbish also. So then you should answer it, question it, and get clarified that yes, the things are correct, or and apply some of the ML technique in your project work, whatever the kind of project was that you are doing. Okay, so thanks. That's it. And maybe next time, whenever I will come, I'll definitely take you through the real code. Just be ready with your laptop, install Python, and install Jupyter, Anaconda, and R. Okay, thank you. Any questions? No. So it was so simple or so complex?
Okay, Avinash, I have a question. Could you please, yeah. So could you please tell us your journey, whether you have solved some industry problem in using machine learning and designed some molecule for pharma company, or some live example if you could share
Yes.
at Grotia and also.
Yes, I have like couple of examples or the case study that I have. So I don't know. First thing is, how many of you you can raise your hand at least? Okay. How many of you want to be an entrepreneur? कोई नहीं, seriously. Two hands. अरे कस के जब बड़ा हाथ उठाओ, so that we all can see that who are the leaders. Only four leaders and sufficient for this crowd, right? So and especially in this domain, like life science domain, it's very challenging, right? Especially when you are going for R&D or saying that, when I started, I said I would definitely discover a drug molecule. That is still my dream. Okay, is not fulfilled. But yes, it is. You can do it. So currently, Grodia is doing this for their clients. Okay, and of course there are lots of reasons, because setting a complete R&D pipeline in house is very expensive. So we thought, that let's start this journey with providing the services to your client, those who are looking for such kind of thing, and then establish yourself and earn money, and then maybe you can establish your own R&D. Okay, so that is the whole plan. Or you raise your investment. So for some reason I decided not now for investment, but later, I don't know, but not now. So so the client that we have currently is of targeting different kind of not diseases, and they have entirely different mechanism. Like somebody is working on small molecule, somebody is working on antibody, and we are helping them to get the quick solution of it. Okay, this is only the work that we can do. Okay, but this is the only work that they are looking for. Okay, like they have, like one of our client have antibody sequences and multiple antibody sequences, and they are targeting some infectious disease. Okay, and they want to know that out of all these 50-60 antibody, which one is going to neutralize this particular pathogen. Okay, now this question can be answered by conducting the experiment, all the 50 assays and all, but then it is expensive for them also. So that's why they came to us and they said that, okay, you just conduct your experiment, computational analysis, and let me know that out of all these 50, which one are the top which can be used for neutralization. So we just suggested them 5, and they tested those 5, and that worked. Okay, so when it worked and they found the activity in the experiment, so what they realized is that they skipped 45 essays, because otherwise they could have would have to do all the 50 essays. And now they also got the confidence ki computational biology is working. So then they came to us and then they say that, okay, for this pathogen you said that these 5 antibodies are working, so can you tell me that for the some other pathogen, let's say pathogen Y, the same antibody will work or not. And the idea was to make a broad spectrum therapeutic antibody. Okay, so and we worked and we are still working on it, but we are not sure whether it will work or not for this next pathogen. But if it doesn't, then you can apply. There are machine learning program algorithm that we are developing, and that will suggest you that what kind of modification you need to impose in this antibody so that it will work for both. Okay, otherwise there are infinite possibilities. You cannot go for all of them. But using this ML approach, we are going to suggest them that okay, just do these things and it will work. Yeah, so this is one example that we have just recently done. Okay, done. Thank you.
Thank you, Dr. Avinash, for such an explanatory session. I may now call upon stage Dr. Manish Narang, who is the director. I would request Dr. Kamal Rawal and Dr. Tanvi Naved to felicitate Dr. Avinash for this session. So now with us we have Dr. Manish Narang, who is a Director, Medical Affairs, APAC at Abbott Healthcare. Sir is an alumni from Jamia Hamdard University. He's a multifaceted professional with more than 25 years of experience in cardiovascular devices and pharmaceutical industry in different functional areas. That includes general management, human financial strategies, medical affairs, regulatory and reimbursement, health economics, professional education. Sir is a unique combination of commercial and scientific acumen and having a proven track record in setting up organizations in Asia Pacific regions. I would like to call here, sir, please, if you could grace us with the session.
Thank you. Thanks, Dr. Naved, for the opportunity, and thanks to you, ma'am, for your kind introduction. I recently completed the PhD from MIT last year itself as part of my professional journey. This was a part-time PhD that I did last year. And I'll start off with some disclaimers and disclosures, which is the first one. I have nothing to do with computational drug discovery, by the way. Right? I have been dealing with human beings for the last 25 years as part of my professional job. I have been involved heavily into clinical research. So out of last 25 years, I would say 23 years, 24 years have been leading and doing clinical studies across different countries. So when Dr. Naved approached me for this course, I was really trying to look for someone who is apt for the topic, but couldn't find anyone. But then I also saw one of the topic was bench to market, and that's where I realized, that's something which I have done. I have been involved in the clinical development of few drugs that have seen the market, as well as 4 devices in the cardiovascular segment that are in the market. So I thought I can talk from bench to market perspective, and I realized I was talking to students, so that also gave me confidence that I know at least few things more than you guys, so I will be able to come and stand in front of you and give my talk. The other objective of coming here was also that I always see these talks or presentations as the practice to the next one that you are going to give. So I try and learn from my fellow speakers, from the audience, from the panelists. So every question that you get asked helps you prepare better for the next question. So that's another reason I decided to come over. Dr. Avinash, I believe he just left. He wanted to ask audience a few questions and there weren't any. So I would forcefully ask you questions. I have been doing this in the past. I have given talks at different universities. I try and make things interactive. So if I come and ask personally a question to you, don't get offended. It's not that I picked you by choice. It's a random disclosure. The second disclosure I want is about my personal life, apart from what was told. So I'm a filmmaker. I produce films. I'm an aspiring actor, still at the age of almost 50. A big Bachchan fan, a sports enthusiast. I own a cricket team for my office. I aspire to be a TEDx speaker. Hopefully before I die, I do want to give one TEDx talk. And of course, I'm a healthcare professional with a serious mission of saving human lives. So let me start with my first question. What is the number one killer in the world? What's the number one disease, the top disease in the world? Anyone can take a guess. Is it cancer? Anything else? Diabetes? Heart disease. Cardiovascular disease was the number one killer 40 years back. It still is number one killer in 2023. More than cancer, more than AIDS, more than accidents, more than anything, cardiovascular disease still remains number one killer, and that's where I thought, coming from cardiovascular background, I feel very proud to be associated with this. And here's a small glimpse of what I do apart from the business of saving lives. And the reason I show this picture is to give you an idea, especially to students, that there is life outside of your professional work as well. A lot of times students think that once they start a job, they don't have any personal life, but that's not the case. As I said, I showed you about my personal disclosure. I do multiple other things. This is the picture from my most recent film, which was released last year. It's called URF, which is based on lookalikes, and it's got screened at various film festivals. I attended to one in Australia and one in Amsterdam, won award in Kerala as well. And the picture below is from 2008 Film Festival in Goa, where I had a 30-second role in that, but the film also won an award. Again, it's not about me, it's about the topic for today, and because we are talking bench to market, I'm going to talk about some of the commercialization strategies of healthcare. So the topic I chose for my talk is synchronizing clinical development strategies with technological innovations. So I'm going to talk about clinical development, I'm going to talk about technological innovations, and do a more of a generic topic, because as I said, I have nothing to do with computational drug discovery, but more about how the development side works. And I start with a quote from Sir William Osler, who was one of the proud founders of Johns Hopkins Hospital, one of the premier medical institutes in the world. And he said, if it were not the great variability amongst individuals, medicine might as well be a science and not an art. So try and understand the meaning of it. You guys heard from the previous two experts on the drug discovery, and we are trying to come up with models that work for everyone. But what this gentleman almost 120 years, 130 years back said, that every individual is different. Even within the family, people have, yes, similar genes and all, but every sibling is different from the other sibling. So there is a great variability in the human life, and that is where the goals of healthcare are, to address those variations, to come up with solutions for those variations, and we'll see how the technical developments can try and support that. So Dr. Shukla spoke about the industrial revolution and how that has transformed the world. Let me take you back 200 years before, where the first industrial revolution happened. She spoke about industrial revolution 5.0 and 6.0. What do you make of this picture? This is from industrial revolution 1.0, almost 200 years back. Any guesses? This is a steam engine, yes, of course. What did steam engine do? Or what is it showing in this picture? Competing with what was then the mode of transport, which is a horse cart. What are the things that you think of with any industrial revolution or any technological revolution? Will it take my job? Right? Will it be safe? Will it be effective for everyone? The first man, who was the first man who stepped on the moon? Neil Armstrong. Did he have fear in his mind? Am I going to even come back to the earth? Of course he had fear, right? But he took that courage and took that first step onto the surface of the moon. So with every technical revolution that you see, there are fears, there are apprehensions, there are aspersions, and people are not sure what that is going to do. But since we have seen 5 industrial revolutions and our Honorable PM is already talking about 6, every change brings new opportunities with it. So it's not that the jobs that were there will not be there. Yes, some of the jobs may be taken, but then you'll always have new jobs. I mean, we heard from Dr. Avinash and Dr. Navjeet on various computational codes that are currently existing, which were not existing when I did my B.Pharm and M.Pharm. I'm talking about prehistoric era, so to say, where even internet wasn't there. So it's not that I ran out of job after internet came, or when we are now talking about chat GPT, which is going to replace medical writers. Maybe, may not be. I think every technology comes with its own opportunities. That's the key message you want to take. So whenever I give any talk, I give a talk with an objective that after 1 year, people should remember maybe 1, maybe 2 takeaways from that. And if that is the case, then my job, my time spending in delivering this talk is worth it. So the key takeaway for you will be whatever revolution, whatever technological innovation may be there, that will give you something which is not currently there. So it's not that you will be out of job with all the technology, you will have maybe something else to do. So now we are talking about revolution 5.0 or 6.0, where we are dealing with robots and dealing with artificial intelligence, and this is the real revolution. What examples you could think of technological revolutions or artificial intelligence around you? Forget healthcare for a moment, but what other technological revolutions you can think of? Again, answers from the audience. Smartphones, of course. What else? Chat GPT. Think of something even more basic. You do online shopping, all of you? Anything you notice while doing online shopping? It doesn't give you choice, right? Whatever you choose first time, next time you go onto that site, it automatically gives you similar choices. I bought something for my dog. The next time I went onto the site, I was filled with choices for that. That's what technology is doing to you. Even the choice of music. You play one song on your Gaana or Spotify, the next time you log on to that, you automatically are given recommendations. That is what is the power of technology around you in very simple words. Again, we are not here to talk about shopping or music. We're talking about innovation in healthcare. Let me share a story with you, which is a story of 2 American presidents, one Republican, one Democrat. President Eisenhower, who was the president from 1953 to 61, and Bill Clinton from 93 to 2001. Representing 2 different parties, but joined by heart attacks. Both presidents had heart attacks in their lifetime. President Eisenhower had a heart attack in 1956 or 57. What was the treatment option available then? Any guesses? Did he undergo bypass surgery? No. Did he undergo an angioplasty, a stenting? No. What was the treatment option back then? Bed rest. Bed rest for 3 months. and of course oxygen, and to relieve pain he was given some morphine shots as well. What were the implications of that? When the news broke out, he was playing golf when he had an heart attack. The stock market crashed. The stock market crashed 10-12% and they had to close the stock market temporarily. What was the loss that resulted because of that? $12 billion in 1958, which corresponds to roughly $200 billion in the current time. Because there was no technology to treat President Eisenhower, he was out of action for 3 months, came back, did another term, and President Bill Clinton, when he was not the president, underwent a heart attack in 2007, was hospitalized, came back 2 days later, resumed his normal life. The world didn't know about it. He had a repeat angioplasty done 3 years later, or maybe 4 years later. He came back the same day. That is how the world transforms with the advent of technology. Yes, he still was a very influential person. He was, Hillary Clinton was still at the office when he had his heart attack. So, significant person, but again, no one got impacted of his heart attack. So, when we talk about healthcare, we talk about the ultimate goal of saving lives, saving deaths. Of course, the improvement in quality of life is there, but you want to live to be able to enjoy the life. So what this graph shows is decline in the death rates over subsequent years, the time when Eisenhower had a heart attack till 2020, where a lot of technology has come, and again, you don't need to go through all these in one day, you're not going to become an expert, but again, any advancement in technology that you think of has to result in eventually saving human life. We spoke about cancer, and a lot of work in genomics is done in treatment of cancer and related disease. So the goal ultimately is to be able to save human lives. We spoke about economics very briefly. I pulled out the list of the revenues of top 10 pharmaceutical companies. And again, my qualification is B.Pharm, M.Pharm, and now PhD, so I can't be dissociated from pharmaceutical world, even though I work for devices. Again, this is, don't go by the source of it, and even if you don't see the numbers, these are the market caps of top 10 companies, which are amounting to $2 trillion. Market cap, not the revenue. So healthcare is an expensive area, very profitable for sure, but very, very important spending from the government side goes into healthcare. What's our goal for Prime Minister? To make India a 5 trillion economy by next few years. So imagine just top 10 companies have a market cap of $2 trillion. So where you guys are going to make a career is in a very, very profitable industry, in a very important industry. So let's go to the definition, because we have spoken about drugs, we have spoken about device. Let's see what definition kya hai. Again, these are there in the internet, so you don't have to worry about it. And I think our previous speaker did touch about the definition of drug. The definition of medical device is not very different. It's an article, instrument, apparatus, or machine that is used in prevention, diagnosis, and treatment of illness or disease, or for detecting, etc., etc. Very similar to what drug does, but it's a device. Can you think of any commonly used medical device? Sorry? ECG, of course, yes. Glucometer, thermometer, stethoscope, these are all medical devices, right? And when I started my career in medical devices, the devices in India were very loosely regulated. Cardiovascular were not at all regulated. There were only 7 or 10 categories of devices that were regulated, but now it's a very regulated industry, even in India. It was a regulated industry outside of India, but even now in India, it is a very regulated industry. So I'm going to briefly cover differences between development of drug versus development of device. But some of the objectives are very common. So when you take any drug or a device, there are a few questions that as a layman come to your mind. When you should take it, what is the dose, how much should you take it, should you take it with food or water, is it toxic, addictive, safe, does it work, side effects, will it make me better, etc., etc. So those are the common principles involved with development of a device as well. This was briefly touched upon by Dr. Ashutosh earlier, but this is the timeline and the development stages of any pharmaceutical product as per industry estimate. When I was studying, the timelines were exactly this. When you are studying, I'm pretty sure the timelines are very similar. This is from a source not too long ago, maybe 2 or 3 years back. The timelines may have squeezed in certain phases, but largely to develop a drug, a new drug from scratch takes anywhere between 10 to 12 years. And when I was studying, the cost associated was $700, and dollar back then was maybe 30 rupees. Now the cost associated could be between 1.5 to $2 billion, and dollars, as you know, is 83 rupees. So any new drug takes a lot of time, and I don't know whether you can see from the back, the lot of time goes into clinical development phase, which is what I do, which is trials on human beings, where you study the drug in different patients, different volunteers, and we'll briefly cover that. Phase 1 allows you to, and this is after you have done the preclinical work, so all the work that Dr. Navtej does and Dr. Ashutosh do, and in the clinical, in the animal labs you do. After that you get to human beings, starting with phase 1, where you test the drug at low doses in healthy volunteers. Stage 2, testing on patients, 100, 200, maybe 300 patients. Then move on to a larger population, few thousand patients, different inclusion criteria. And once you collect all the data, give it to the regulator, the drug is approved. We move to phase 4 stage, which is post-commercialization, post-marketing surveillance, or whatever you may call it. This is a graph from the world map showing the number of phase 4 studies done worldwide. You don't see the number here, but these are close to 2 lakh studies done at any point of time world over in the phase 4 arena. Because when you study a molecule or a device, it is studied in a limited indication. In case of drugs, say 20,000 patients, 30,000 patients. In case of devices, I'll talk about it, it's even lesser. But when it comes to use, how many angioplasties are done in India currently? More than 10 lakhs. How many devices have been studied? Maybe a few hundred. So real worry lies in the post commercialization real world data, where you study the drug or the devices in different populations. Because I do clinical trials, I thought let me briefly talk about types of clinical trials. So trials could be industry initiated, which are funded by the industry, could be government initiated, like the ones which are initiated in the COVID period, could be academic trials. A lot of government institutes as well as private institutes run their trials, and a lot of investigator initiated. So once the product is commercially available, investigators, who are the real connect between the developers and the patients, they do their own research as well. And in terms of type of studies, it could be looking at a treatment, prevention, diagnosis, screening, or evaluating the quality of life. And again, whenever you look into any trial, which I'm sure you look at a later stage in your career, look at how good that study is on the basis of whether it has a good trial design, how well is the trial conducted, and what was the reporting mechanism of the study. Now, this is an important slide because it talks about difference between the development requirements and cycle of medical devices versus medicines. One thing I forgot to tell you earlier when I was giving my introduction is I am very used to the word cut. So whenever you think I'm not making sense or you want me to elaborate on anything, say cut. Okay, I forgot that, but yeah, I certainly might remember that. So when we talk about medical devices, the development cycle is not as complex. It doesn't take 12 years to develop a new medical device. The development cycle is much small, and it's a continuous process, and I'll give some example later on. It's a cyclical process versus a drug compound where once it is approved, you don't make any further changes in the same molecule, you only develop a new molecule. The preclinical is also short, and the clinical trial requirements are always not there, whereas in case of drug, you always have to undergo a mandatory clinical trial phase. Different phases are there. You start with initiation, looking at the opportunity and look at the risk. Obviously, you compare yourself with what is currently available in the market and accordingly decide whether you want to put in your money in developing something new. Start with the formulation, concept, feasibility, then start working on the design development, and then do a verification and validation of a prototype, and then come with the final validation and eventually launch the product and do the post-market assessment. Theory remains similar to what the drug development is, but certain changes in terms of scope and all. So I work in an organization which develops a lot of medical devices, cardiovascular devices. It's great to see a new device working in a patient, and it's very rewarding for all the work that has gone into. And then subsequent trials that are done to mainly confirm your theory and all. And always ask yourself whether the trial is really necessary, because it takes a lot of time and effort to do any study. So what you're trying to accomplish is basically confirm your proof of concept, demonstrate that your product is effective and is also financially viable. You don't want to develop something that doesn't have a market. Remember, 2 trillion from top 10 companies, that is the goal of a pharmaceutical or a medical device company, eventually to make money. And once it is there, you talk to your thought opinion leaders and run it by them and all. So this is an example from one of the products that my company made. It's a cardiac stent. I don't know if you know about stent or not. It's implanted in the arteries to clear the blocked vessel. Any guesses what is the size of an artery in terms of the diameter? Make any guess, any number. Okay, what is the unit? Forget the number. What's the unit? Is it centimeter, millimeter, inch in terms of diameter? Millimeters. So it's millimeters. So an average artery is 2.5 to 4 millimeters. Indians have a smaller artery. So remember, we are operating in the tiniest of space. And you do the operation while the patient is still alive with an objective to keep him alive and improve his life. So this is the development cycle of one of the products. We started with the first in man way back in 2004. Went on to do a larger study, 300 patients. And we got the approval right at this stage. Right at the stage where we had this 300 patient data, we got the approval to implant the device and take it to the market. US approval was done on the basis of 1,000 patients in these 2 trials, and these were completed by 2007. Subsequently, 16 years later, we still continue to do more and more and more work, trying to develop different generations of that product, as well as generate data which can effectively give the physicians an informed choice. If you want another takeaway from my talk, this is the second takeaway. First, I have already given you, this is the second takeaway. In case of devices, you don't necessarily see radical innovation. So what's the radical innovation here? From a landline to mobile phone was a big radical innovation. From a mobile phone to a smartphone was another radical innovation. But from iPhone 3 to yesterday's iPhone 15, God knows how much are we fooled by Apple for maybe 10 extra megapixel or some extra storage. We end up paying hundreds of dollars to Apple just for that. The basic feature remains the same, with obviously some innovations and all. So in medical devices, you don't always see radical innovations, you see incremental innovations. We make incremental innovations in our product as well. I showed you the life cycle. I mean, it's not that we are fooling the doctors or the patients as well. There is some science that goes into it. But yeah, those large innovations are not always there. Again, I'll close the medical device topic here. So when you develop a prototype, make sure it is safe, make sure it's built by engineers, it is used by key opinion leaders with guidance from inventor. We saw Dr. Avinash spoke about his invention and his journey. And when it comes to actual commercialization, you want the product to be safe on your grandmother. That's a test every company always does, that if I'm developing a new product, testing it on patients, I want to be safe in every single patient, including my own family. And it's going to be used in a very real world clinical situation. So it has to be working every single time and must be cost effective as well. Again, the idea is to practice evidence-based medicine. This is a pyramid where real world studies, randomized clinical trials, meta-analysis, all those help clinicians make an informed choice to treat the patient with an objective to eventually save lives. I'll very briefly in the next few minutes talk about innovation goals, since a lot of you will be involved in research in the coming years. Let's talk about innovation in non-healthcare space. Again, I'll run this video, but what do you think of innovation in non-healthcare space? Think of automatic cars. Why is this video not playing? Anyways, this is an automatic car, a driverless car, sorry. When is it going to hit the market? Okay, let me rephrase the question. Will you be willing to drive a driverless car? Yes? Someone is nodding yes? No? It's going to hit us very soon. I mean, before we realize it is going to be there, the prototypes are already there, tested and all. So these are going to be very soon. So when we can accept a driverless car, being able to check my sugar when I am talking to you should be straightforward, isn't it? So while I am talking to you, I am checking my glucose. That's a goal of innovation in healthcare space. I don't need to prick. I have a sensor attached to me which should give me an ability to check my sugar to see whether it is rising, falling or whatever. That is how innovation in healthcare is progressing. Everyone has seen Apple Watch. What does it do besides telling you about your steps and all? Also gives you your heart rate. It can alert your doctor in case you are having irregular heartbeat. There was a study conducted and presented a few years back using Apple Watch. It was done in more than 4 lakh patients. For a normal study to be done without Apple Watch, it would have taken easily 10 years to get 4 lakh patients in the study and millions of dollars. All this is done while people are doing their daily work, and that is the goal of innovation in healthcare. We talk about smart cities. In healthcare, we talk about smart device which could give you smart care, eventually giving you a smart home. Forget smart cities, we are talking about smart home now, where everything is wearable and your data gets transmitted to your doctor, you get care right at your doorstep. And this is not future. I told you about glucose level right away. I don't wear Apple Watch, otherwise my heart rate would have been known as well. And now let's talk about artificial intelligence, again, which is not my subject domain, but I gave you a few examples of what artificial intelligence does, which is, in case you have to understand again as a layman, maybe the third takeaway, it's all about processing the data. Imagine it as a super Excel. You work on Microsoft Excel, right? Imagine it as a super Excel which can handle large volume of data. You may not like my simplistic explanation, but because I'm not a subject expert, that's how I simplify it. Any data that can be analyzed becomes part of the source of the data. And importantly, we spoke about machine learning and deep learning. It gives you an ability to predict on the basis of data. Now, you go to a doctor, one doctor charges 3,000 rupee fee, the other charges 300 rupees fee. It's also a function of their clinical experience. You go to one of the best doctors, how and why is that doctor best? Because he is able to diagnose much better, much accurately, much quickly than someone who's just come out of the medical school. Which is what the goal of artificial intelligence is, to be able to predict in a quick time, in a realistic fashion, very specific, very sensitive, on the basis of data. How do doctors become expert? Is by treating patients, by seeing more patients. Anyone has an exposure to a government hospital? Again, nothing against government hospitals, I work with them a lot. Anyone been to AIIMS? GB Pant? Anyone been to OPD of AIIMS? How are patients treated? How are patients patients treated in the OPD. Lot of crowd. So the doctor is holding one patient's pulse, he's checking one patient's pulse, talking to second patient, writing prescription for a third one. And if you do that day in, day out, I'm sure your expertise is going to be perfect by the time you complete 5 years of residency in AIMS or GB Pant or any premier government institute, vis-a-vis a private institute where you may not see as many patients every day. So artificial intelligence basically helps you predict that. Again, it's a very scientific thing which I am not an expert on, but yes, there are machine learning modules and deep learning modules, and now I see school students doing that as well. So college level, I mean, you guys should be master at it, as Dr. Ashutosh helped you understand it earlier. Again, it's nothing new. It has been there in the past, so you can Google it. You'll find a lot of information on history of artificial intelligence. And some of the examples of artificial intelligence, there are robots, and where they help in, maybe I should play this video. This is a robotic surgery being done, not on a patient, but on a seed, on a grape actually. Imagine, can a human perform a surgery with this precision? No. The best of surgeons can't move their hand 360 degree. The best of surgeons can't do that. Robot does that for you. And nowadays robotic PCIs are done, robotic angioplasties are done where the physician is not even there in the cath lab. So, do I have a few more minutes, or I'm happy to wrap up whenever you want. Some of the examples of artificial intelligence in healthcare. Oh, why I'm going back, sorry. Now, why apply artificial intelligence in healthcare? What types of data should be analyzed with AI? What are the applications that enable AI to generate more clinical results? And what are the diseases which are currently managed by AI community? Cancer, neurology, cardiovascular. Again, these are the top killers in the world. Cancer, cardiovascular, we know. These are the ones which are currently managed by artificial intelligence. And what artificial intelligence in healthcare relies upon is a lot of patient data. So there are hospitals who sell the data to these companies managing artificial intelligence, and based on their own data, they come up with a software, they come up with an algorithm to provide more treatment of algorithms to the physicians. It's a cyclical thing. All of you would have seen various apps on your phones which track everything that you do on an hourly basis, and that is what the role of artificial intelligence is in healthcare space. You have 3D printing, you have less invasive diagnostics, patient-facing mobile apps, remote sensoring. I was at a conference in Amsterdam a couple of weeks back. There was an entire section dedicated to digital monitoring, because in COVID we saw the importance of digital monitoring and how that can help improve the patient interface with the physicians. When we talk of AI in drug discovery, we talk about improvements in clinical trial design, helpful in ensuring quality assurance and quality control, manufacturing, management of the products, as well as drug screening, which has already been spoken about. So this graph on the left again shows you the cycle of drug development, and on the right the chart shows you where AI driven protocols can make the clinical trials more intelligent, because this is where you end up spending most of the time, end up spending most of the money. So with AI you can improve this and run better trials and try to reduce the time cycle from 10 years to maybe 8 years. Even that is a huge saving for a company like us. Again, all this is there in public domain, and there are different sites which show the example of AI tools used in drug discovery. You can go through this whenever you have time to educate yourself. And I guess I'll end up my talk here. Again, AI in healthcare is no longer new. I said in one of the meetings earlier, a few months back, that by the time people start thinking of doing something in AI, who knows, AI may replace itself. So don't waste the opportunity, make most of your time. AI is there, and it's best to be friends with it and best to understand it as much as we can, because there are a lot of opportunities already out there. With that, I'll go to the last slide, which is again taking cue from the first one. The great variability amongst individual makes medicine an art. So Dr. William had this quote about variability, and that's where he says he knows, he acknowledges the variability, and that is where medicine is an art and not just a science as per him. Thank you very much.
Time for a few questions. Audience.
I didn't hear any cut, so maybe you understood it all.
Maybe you forced them, as you promised that you will force them to speak up.
Yeah,
also.
yeah.
Thank you, Dr. Manish.
Thank you.
Dr. Manish,
There is a question, sir.
Yeah.
So please go.
You'll have to speak a bit louder. Yeah.
Can we pass the mic, please? I request you to introduce yourself and then ask, please. Okay.
Good afternoon, sir. My name is Bushra Khan, and I am from Jamia Hamdard too. And my question is, since you have been a part of the development process and you know how the things work, so where do you feel like we have the technology, we have human resource and everything right now, but where are we lacking in the process of developing drugs which we could speed up and it could be more effective? So where do you suggest we could work on?
So if your question is hinted at, why don't we see basic drug development in India, right? Is that your question?
Sir, to be more precise, my question is how we could improve our process on development of drug and reduce the time that is taken.
So I think the experts before me already spoke about that in trying to reduce the drug discovery cycle. I think drug development cycle is something which, because of regulatory, so whenever you take any drug, remember the few concerns that were there, whether it is safe, effective, it works every time, etc., etc. So whenever regulatory body approves any healthcare product, they got to make sure it is safe for the larger population, it is effective for the larger population, and it is better than or equivalent to something which is already there, which is where the bulk of the time is spent in testing it in first in lab, then in animals, then in human beings. Ultimately, it has to be taken by human beings. So even though you do a 10,000 patient study, it is used word over. So you want to be sure scientifically, statistically, theoretically that it is going to work every single time. There have been cases where the drugs have not worked well and companies have paid billions of dollars as compensation. I mean, there are n number of examples where an approved product hasn't worked. Forget about those which were in testing phase. Even the approved products have had side effects which are not known at the time of development it, which is where the governments, the regulatory bodies, and even the companies, because I as an organization would not want to pay any single dollar out of my earning as a compensation. So I would want to develop something which is very, 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 very safe and effective. That's where it does take time, but again the efforts are there to reduce or to make our trials more effective. Now with all the machine learning and all, whenever you think of a trial, you already know which are my top 20 hospitals to go. We recently published a study at a conference in Amsterdam. It was done for about $40 million. It still was a negative study. It's $40 million out of our pocket, but it's a negative study despite using the best of brains and all. So you don't want to use the money in a non-profit direction. That's why it's a lengthy process. I mean, in case you want to add anything on reducing the timeline, I think efforts are obviously, no one wants to wait 10 years to make money. You want to start making the money from if possible, day one. Thank you. Thanks for the question.
More questions?
Thank you very much for enlightening us about the drug development process and the regulatory affairs. So, in one of the slides, you mentioned that you got the approval in phase 2 itself, and the US approval in phase 3. Why is it so?
So in that case, I mean, because of lack of time, I didn't elaborate. The approval was for outside of US. US approval came only after we did those two studies. But for markets like Europe, Asia, the approval was after phase 2. Again, in devices, we don't necessarily have a phase 3. In drugs, we need to have a phase 3. So the number of patients or healthy volunteers tested in phase 1 in drugs is typically 10 to 50. And those are more on healthy volunteers, and pharmacokinetic studies are done. Phase 2, you actually take it to patients with about 100, 200, maybe 300 patients, and you do maybe 2 or 4 phase 2 studies. Then you expand it to more sites and more number of patients. So in phase 3, it typically starts with 1,000 patients. Then the next study could be a 5,000 patient. Then the last study could be 10,000 patients. One of the product I was involved in phase 3, which is there in the market now, underwent almost 25,000 patient study in a total of 4 or 5 studies, and that went on from 2008 till 2014 before the drug eventually was approved by FDA. It got rejected once. We had to do another study and generate more data. So phase 3 in drugs is much wider, much longer. Phase 3 in devices, there is an overlap between phase 2 and phase 3. I mean, there isn't a real phase 3 in devices.
It means that US is using the more stringent criteria.
Well, you can say it is stringent, but again, US studies in case of cardiovascular devices is 1,000 patients only. Outside of US is 300. So does 1,000 make it more stringent? I don't think so. It's just that US FDA wants it to be 1,000 patients, and European Union and Indian government allows it in only 100 patients.
Right.
So,
and my second question is related to the COVID-19, where the remdesivir has got the US approval, FDA approval, but later on it was found that it is not acting against the COVID-19 itself, SARS-CoV-2 virus. So how the FDA, this drug has got the FDA approval?
So the
although it is not acting against the SARS coronavirus.
So COVID time was very different. Again, we need to understand the requirements of COVID. In most of the world, including US, it was largely hit and trial. People knew this is an antiviral drug, and even people tried something which was very well known, like a hormone, right? So a steroid basically. So whatever was they thought is going to work, people tried that. It was only that later they analyzed the data in more details and found that this is not the best treatment and has more side effects.
But it's the first criteria is the drug should act against that particular target, right? That is in the case of remdesivir, SARS coronavirus. They might have seen the data.
They did see the data, and based on the expert judgment back then, they would have approved it. But again, COVID was a very different time and is not going to be the norm going ahead. I'm sure. Hopefully, we don't undergo another such.
There were many things in the COVID. Yeah. Thank you very much.
I think I would like to add the COVID was emergency use authorization was provided. So it was not on stringent data and the routine FDA parameters which are needed to be taken care. The standard has to be met. Nothing of that was followed. Even the vaccine came in 1.5 years. A lot of vaccine got failed.
That was entirely different scenario.
And even the vaccines were approved for emergency authorization.
Yes, it's EUA. It's a normal scenario. They might not have met those criteria which are there in.
Absolutely.
Yeah.
Okay.
Thank you.
Any more questions? So I would like to thank the speaker, and he has mentioned the way forward is artificial intelligence, whether you are doing drug discovery, whether you are developing new medical devices. I think he has rightly touched upon that aspect, and probably, he is doing a great job in introducing AI. He is aware, and he is, putting those technologies in picture. And we'll be happy, because we are very strong in artificial intelligence, especially in healthcare domain. And we have been executing projects for pharma companies where we have developed new technologies based upon funded projects, and as well as helping a lot of corporates and also.
Great.
Right?
To know that,
Thank
yeah.
you.
Thank you.
Thank you very much. Before we go further, we would. Yeah.
Thank you, Dr. Narang. Such an exhaustive, such an entertaining session with our students. I hope everybody must have enjoyed and learned. So may I call Dr. Tanvi Navid sir and Dr. Kavit Raval sir to felicitate our eminent guest, Dr. Manish Narang sir.
I would also like to invite for this felicitation Dr. Mukesh Churasia. Please come.
Thank you so much, everyone. At the end, I would like to call upon stage Professor Dr. Kamal Raval sir. He's a center head, Center for Computational Biology and Bioinformatics, Amity Institute of Biotechnology. Sir, could you please say a few words?
Thank you very much. Ladies and gentlemen, my dear students, esteemed speakers, my colleagues, good afternoon. I stand before you with an immense gratitude and heart full of appreciation as we conclude this enlightening workshop. It has been an enlightening journey through the realms of cutting edge sciences, and I would like to extend my sincere thanks to all those who have made this event possible. First and foremost, I would like to express our deepest appreciation to our distinguished speakers and presenters. Your expertise and insights have illuminated the path from the lab research to practical drug development. Your dedication to advance the field of computational drug discovery is truly impressive. Specifically, I would like to thank Dr. Navajit from Redcliffe Genetics, and he has discussed about genomics, IT, and other initiative at his company. Dr. Avinash Grodia, who was a lead scientist, he has discussed about basis of drug discovery. He has discussed about limitation of docking, emphasized the need of machine learning in the drug discovery domain. Then Dr. Manish from Abbott had shared his experience to bring drugs and devices to the market, and he has also emphasized about the commercial aspects of drug development, because if you do everything in the lab and research, it doesn't come out and help people in the society. Your research has limited kind of application in the for the world outside, and of course the investment won't come. You won't be able to pursue your research further if nothing comes out of your lab or your research activities. And he has rightly mentioned about AI, and I would like to thank our Honorable Vice Chancellor for sparing her valuable time and sharing her expert thoughts on different aspects of drug development. Dr. V. Pooja, officiating head, Dr. Puniti Mathur, my colleagues, Dr. Naved, Dr. Annie, our partner institute from AIP, the volunteers who have worked very hard, and Dr. Mukesh Churasia, Dr. Jitender, IT team, hospitality team, and administration for making this event possible. Thank you very much. And we need to, the participant have to fill up one quiz and very short feedback form. The links will be shared soon. And probably I'll just, okay, please note it down on a piece of paper or you can do it on your phone. It's a Google form, only few questions. tinyurl.com/. CBAI, C for cat, B for Bombay, A for Apple, I for India. CBAI, that is a new name for our center, Center for Bioinformatics and Artificial Intelligence. So CBAI 1000, 1000 numeric, and just typing it on the. This is the link. I'm just going back. Is it visible? Is it visible? And the second link is over here, 2000, 2000. आप भी आ जाओ, आप ले लो।